No.
All right. Thanks everyone for joining. It is really my great pleasure to introduce the talk for today as I'm presenting Alexa, my great colleague who has been with us at the Douglas for a while. So it's really my pleasure to introduce Alexa Piché-Binet, who obtained her PhD in neuroscience from McGill University under the supervision of Dr. Sylvia Villeneuve. She then moved to Lund University, Sweden, for her postdoc, where she joined a group of Oscar Hansen and the BioFinder team. Her research integrates multimodal imaging and biomarkers to better understand aging and Alzheimer's disease. So as Alexa has mentioned, feel free to ask any question during the presentation. Just to keep it, I guess, a bit more orderly, if you're on Zoom, you can raise your hand or ask in the chat, and I will find a good moment to interrupt Alexa so that you can ask your question. If you're in the room, I will try to keep my eyes open for hands raised, but I'm not sure I can see anyone. So Malav, feel free to jump in as well if something's missing.
Yeah,
So
sure.
without further ado, Alexa, I'm very excited to hear your talk. So the floor is yours.
Thanks, Fred, for the introduction, and thanks for inviting me. So I've benefited from this lecture series when I was a grad student, and now thanks a lot for inviting me as a postdoc, even though I went away. And maybe, Malar, I could have told you 2 years ago, but thanks so much for continuing this series. I remember when the pandemic started, but still it kept going virtually. And I remember it was very nice when I was still finishing my PhD to be able to take part in this and still feel the community. So thanks a lot for that. And so today I will talk a lot about Alzheimer's disease, PET imaging, but also novel biomarkers that we're starting to investigate in Lund in Sweden, as Fred mentioned. And I know you've had many nice talks about Alzheimer's over the past months or years, so I won't bore you. A big explanation on Alzheimer's disease, and also a big thanks to Valent last week, who really gave us a really nice overview of the disease course, so you can catch that when it's online. But for the talk today, I think it's just really important to remember that we can really see Alzheimer's disease as this clinical pathological entity. So of course, we know the clinical symptoms, with often memory that is being affected first, and it's a progressive decline that will lead to in the late stage of the disease when the activities of daily living are also impaired. But before there's so many pathophysiological changes that starts to occur in the brain decades before there's any symptoms. And they're characterized by the presence of extracellular plaques of beta amyloid. Intracellularly, we have these accumulation of fibrillary tangles from tau proteins, and there's also neuronal loss. And I will focus very much on tau today, so I also really wanted to make sure that we're all on the same page when we think about tau pathology. And the protein coding for tau is this microtubule associated protein tau, so MAPT. And tau really helps to lyses the microtubules in the neuron. And first, they will get hyperphosphorylated and then starts to accumulate in monomers and oligomers. And this is the soluble form that we can measure. And then over time, these aggregates, and we get these Larry tangles. And this is really the insoluble part. And we have different biomarkers for these different phases of tau. This is also true for amyloid, that has this soluble and insoluble. So this will be important when we talk about different biomarkers over the course of the talk. And we're very fortunate in AD in terms of being able to measure a host of biomarkers. So of course, we have PET, as you know, I think, to look at amyloid and tau. And with PET, we're really looking at these end stages, these insoluble aggregates of tau, or these plaques of amyloid proteins. But we have also more and more fluid biomarkers to capture this. So yeah, this soluble part that I was talking about. So we have proteins that we can measure in the cerebral spinal fluid, but also more recently in blood. So now we really have a host of really less invasive biomarkers that are still in research at the moment, but are getting very, very close to the clinic. So I think that's a quite exciting area that we're in the field of Alzheimer's disease. And especially in Lund, there's this big expertise in a lot of these novel fluid biomarkers. And so today I'll talk about one of them that is particularly interesting. And in terms of biomarkers, also, we're reaching a new era with more and more omics data that are becoming available. So we can really start to investigate different biological processes. We're not limited only to amyloid and tau. Of course, the disease is much more complex than this. And I'll end with some ongoing work that are mostly focusing on, and this will be still more new, so I'll be very happy to hear your thoughts and discuss what you think about this part. Let's first start with some more PET data. And really, in the field, there was this big debate as to what it entails, let's say, when someone who's still cognitively normal, but has amyloid and tau in their brain. Is it just simply related to increased risk of AD, like age or sex is, being female is higher risk, for example, or is it really very closely related to clinical progression? And this debate, if you will, really stemmed a bit from two different criteria that we had in the field to view the disease. So we had this more, say, biological first definition where, and sorry, just to say here, this debate really that I'm depicting here is really focusing on these cognitively unimpaired individuals. So before there is any symptoms. And so according to this biology first definition, we could see that, so not having amyloid or tau is normal biomarkers and not at risk for progressing to AD. But according to our blue definition, if you have amyloid only, so A plus T minus, this is already called preclinical AD change. And when you have both pathology, then we call this really preclinical AD. But when you look at this other definition from the International Working Group, that is more syndrome first, you see that regardless what you have, it's just amyloid or both pathologies, you're still always just at risk for progression. So we really wanted to see which one of those definitions is really key. And amyloid PET has been around for a long time, so since 2004. Tau PET is a bit more new, around 2015, 2016. So we still have many cohorts that have this data, but we still needed a few years of cognitive follow-up to be able to know if to have cohorts that people who will progress or not. And so it's only since recently that we've been able to address this question. And how we went about it was to try to take advantage of a lot of cohorts across the world that have baseline amyloid and tau PET, and also cognitive follow-up, as I mentioned, 3 to 5 years, to be able to have some people that did convert to either mild cognitive impairment or dementia. And so we grouped all these data from our nice collaborators that agreed to share with us to have these 4 groups that you have shown here. So you see the vast majority of participants, they were negative on both amyloid and tau. We have this group that was only having amyloid in their brain, so A plus T minus. And this is the region we looked at for amyloid. This is really this neocortical amyloid score. This is really common way to look at amyloid. Amyloid is very widespread already in the preclinical phase of AD. And for tau, we split our groups into 2 different groups. We have first a group with more restricted tau, only in the medial temporal lobe. And this is really the first regions where we can capture tau with PET. And so these are A plus T plus MTL. And we have a group that is a bit more tau extended in the neocortex, specifically in the inferior temporal and middle temporal. And from all the PET studies now, or neuropath data, we know this is really the next regions where tau is going. So tau has really this pattern that spreads way more with the disease. And so these participants are a bit more advanced in their tau, if you will. And you'll see, I think it's also very striking, right, that these groups are also relatively small to find in cognitively unimpaired individuals, so about 10% of them. So I think it really speaks to this importance, or this nice approach of pulling the data together to be able to look at these 4 groups separately. And so the first question was to look at differences in clinical progression between the groups. And on the left, you have the survival curves when it comes to progression to mild cognitive impairment and to cause dementia on the right. And I think it's very striking already that it is really our 2 groups that are A plus T plus that had the greatest chance to progress to mild cognitive impairment. Again, this is over 3 to 5 years in our sample. And so hazard ratio of, almost 15 for the group that is more restricted tau, but the one a bit more extended in the temporal lobe, almost 20 times higher chance to progress compared to the negative group. And also these are also highly significantly different from just having amyloid in the brain. And so after 3.5 years, there's about like 50% in both of these groups that progress to MCI. And if we looked at this time, here it is really only the group with more extensive tau that had the greatest progression to dementia. Very high hazard ratio, 41, but here also let's just keep in mind that this group is relatively small, right? Over a few years, there's not that many people that converted to dementia. But still very clear that those A plus T plus are really the one progressing. And then we looked at the data a bit in a more, slightly, I cannot speak, it's so bad. We looked at cognitive decline. So in a similar way, but instead of progression, this is looking at cognitive decline. And on the left, you have decline on the composite score that is meant to capture really early changes. So preclinical Alzheimer's composite score, it's called. And here we see that both groups are showing a steep decline compared to either the controls or having just amyloid in the brain. But when we look at the MMSE, so a much more global score, this time reflecting a bit as we saw with the progression to dementia, it's only the group that have more extensive tau that shows decline. And we wanted to dig a bit deeper into this. So this MPAC score is actually made of it includes this MMSE, but also 3 different cognitive domains that you see represented here. And when we look at memory here, as we saw on the global score, both A plus T plus are declining. So even if you just have tau restricted to the medial temporal lobe, you can already show signs of a memory impairment. But on these other domains, timed executive function or semantic memory, then this is only the one that have more extensive tau that are declining. And also, if we did a lot of sensitivity analysis here, like making different groups based on the different age ranges or further adjusting, let's say, for white matter lesions, and all results remain quite strong. So really, I think strong evidence for having both amyloid and tau being related to progression. And if you know the AD literature, or probably this has been shown at the Douglas before, this was a very hot question, right? So, there's a question I can answer right now if you want.
Thank you. So for the delayed memory, the startup, the baselines were the same, because in your graph, they look like they have already started to decline.
Yes. Yeah, that's a good point. And I would have to check again, but I think it was trend level or very close, but these groups were a bit small, and not small, were starting a bit lower. And I have to say it was not exactly the same test across all the 7 cohorts, but it was still very highly comparable. And in the supplements, we also have the results for each cohort separately. And I think there we see also some heterogeneity. Some cohorts are, declining more than others. So I think it's also worth keeping in mind that this is pool data, but you can look into more details also by cohort.
Alexa, while we're here, can I ask one more question? Is that okay?
Yeah, for sure.
So how did you tease apart these 4 biological subtypes here? I think you may have mentioned it earlier and I may have missed it. How did you actually was it data driven or just based on observation, or? And
this is all based on PET. So I think if this was clear.
But it's clear that it was based on PET. Just the 4 subtypes, or were they defined based on previous literature or based on
the literature, or what we know about the pathology so far in general? So you might notice that we don't have this A minus T plus category, for example. So this one is very small. This is also some debate. We can discuss this. We can come back to this category later if people want. But in general, it's very, very rare to have extensive tau without the presence of amyloid. So this is why, let's say, this category is not there, and we only have the A plus T minus. And then for the tau, how we split this in these 2 groups, this again, from now years of tau PET data, it's very clear that these are really the early regions, and these next more lateral temporal are really the next one. This is very clear. So I think part literature, part what we know from the data from the recent.
So was it data driven from this data set, or these thresholds were defined before? So
so for the threshold, what we did is for the amyloid, this is because it was a bit different, and each cohort gave us, we set the rules, let's say, and then they gave us their category, who's like positive or negative. So for amyloid, because this has been around for so many years, I think each cohort, they really have this easy way, or established way, sorry, not easy, but established way to say who is amyloid positive or negative. So from each cohort, because there's not always the same amyloid tracers and so on, so this we relied on them to say like, okay, we know who's positive or negative. And amyloid is showing a very bimodal distribution. So it is very easy, again, maybe not easy, but we can, it's very, with, let's say, just Gaussian mixture modeling, if you will, we can really well separate who's positive and negative. So that was for the amyloid. For the tau, it's more tricky. But what we did here is, to be positive, you needed to be 2 standard deviations above the mean of cognitively unimpaired amyloid negative people. And those really don't have tau. And this also, let's say, internally with the BioFinder study is the one that we have in Lund. This threshold also matches very well when we compare to Gaussian mixture modeling and so on. And it's a very common way with PET data to establish a cutoff of positivity. So this is 2 different ways that we went for the amyloid and tau, but they really make a lot of sense with what people are doing in the field, what we know from having worked with the data. So that's how we went to try to have something equal across all the cohorts.
Sorry, one last question. I'm curious about this. So the yes, no for A beta, or I guess the same for the tau, is it done at the voxel level or an ROI level?
ROI level. So for the amyloid, it's really the average across this whole neocortical region, because this is really where amyloid is. It's like very, very widespread. Whereas for tau, it was this yes, no based on the average from this, more restricted RI. So either only in the medial temporal lobe, which was actually entorhinal and amygdala, or rest of the temporal lobe, which was inferior temporal gyrus and middle temporal gyrus.
So I think we have one more question.
So, have you tried to look at it in as a continuous variable rather than grouping them?
Not here because of the cohort differences. Even with the tau PET, there's some, again, not all the tracers are the same. And I think we're not quite there yet in terms of harmonizing across all the tracers. So we didn't do it. But for example, again, this BioFinder cohort is the one I know the most now. It's very large. And if we look at there's many associations with just tau levels and cognitive decline. This has been quite well established. Tau is tracking way more closely the cognitive deficits compared to amyloid.
So that was exactly the question I was trying to ask. Is it a threshold effect that you pass it and then you see the hazard ratio jump to 15, or is it like a dosage effect? As it's going up, you see more and more. Like that was basically what I wanted to ask.
That's a really good point. And I think I think there's a dosage aspect, but with tau, there's also a localization aspect, right? Because it's like spreading a lot throughout, and we'll I have lots of tau data, don't worry, I like it. But it's spreading. So I think this difference that we see actually between the more restricted and not so much more, right? It's just a bit more in the temporal lobe, but I think it's already striking where, memory deficits could already be present there, but to have more global deficits, then the tau needs to spread a little bit. So tau has this, I think, nice aspect about it, and makes it a bit harder to work with, right? Which ROI do you decide? Because then it can be different by people. But I think dosage and localization for tau. Great. Should I continue? Perfect. Thanks so much. I think it's more fun to discuss at the same time, so don't hesitate. And as I was saying, and now we've seen how it's a hot topic, we've just discussed this for a few minutes, but there's also very matching evidence coming from the Douglas, obviously. So different cohorts treated separately this time But I think we see the results are very similar, right? So we have the A plus T plus group that I've put in the red boxes. And again, it's very clear how this is really the group that is progressing the most. Again, about 50%, sometimes even up to 80% of people progressing over a few years. And here, a nice thing that Sherry did was also to add a measure of neurodegeneration. So the groups become smaller, of course, but if we now look at those triple positive people, let's say A plus, T plus, N plus, again, in the red boxes, now we see the rate of progression goes up in most cases, and sometimes from up to 70 to 100% of people that are progressing. So I think, yeah, even adding a marker of degeneration, maybe we can get a bit closer even to the chance of a clinical progression. So I think these two studies together, they really are a strong evidence that the presence of amyloid and tau, also tau a bit more in extensive regions in the cortex, are related to cognitive decline over a few years. So really supporting this preclinical definition that I was referring to in the two different criteria. And I showed you a bit at the beginning, the pathology of tau is more complex. And before we get to these insoluble aggregates that we measure with PET, we also have these more soluble aspects going on with increased phosphorylation and the release of soluble tau. And another nice aspect also, now that we are starting to have more and more tau PET data, is also to be able to look at the accumulation of the pathology over time and really try to understand a bit better all the mechanisms and also what might be the best biomarkers to be able to track the accumulation of pathology. And one of those markers is p-tau217, so phosphorylated tau 217, so this phosphorylation at the epitopes 217 of the protein. And this one has been a key markers that they've developed here in Lund with collaboration with Eli Lilly in the recent years. And so we're really interested to understand how such markers then are actually related to the accumulation of pathology and also cognitive decline over a few years. So try to understand a bit better our pathophysiology of the disease with in vivo data and especially using a longitudinal data. So to answer these questions, we used this biofighter study that I mentioned in the first project that I showed. So this is a really large cohort that we have in Sweden, and people are followed for up to 10 years at the moment. Most people are reaching the 4-year follow-up, and they are very well characterized with lots of biomarkers. And so for this project at baseline, everyone had the CSF p-tau, so our soluble measure of p-tau, and an amyloid PET scan. And for longitudinal measures, then there's multiple tau PET scans and also a multiple cognitive assessment. So those two over 2 to 3 years. And we extracted the rate of change for both longitudinal measures from the slope from a linear mixed effect models. And you see all the brain results that you'll see are generated from SUVR on the Schaeffer Atlas, so 200 brain regions. And first, I just thought to show you the tau PET at baseline. So on the right, you have these amyloid negative controls, and they are very, very little tau. So it's really the people that are already have amyloid in their brain, a bit as we saw before, that also have tau. So here, very little tau. Up in the middle, we group together the controls in MCI who are amyloid positive. And you can really see those are the ones showing the highest level of tau. Again, this very nice uptake in the temporal lobe, as we expected. We saw this is the early regions. But then also the next regions are more parietal, lateral parietal, medial. But also it's really in the AD dementia that then the levels of tau PET are very high, as you can see. And then mostly important actually for this project is rate of change. So this is tau PET SUVR change over 2 to 3 years. So SUVR change per year. Yes, another question.
I'm curious about the test retest data for each of the SUVRs per region you're exploring. I mean, are you following up on Is that your control the A beta negative? What's the control for the test retest?
The controls here are just, this is not really control, but cognitively unimpaired, amyloid negative. In terms of, we didn't do specifically test retest, or do you mean to calculate the rate of change, or?
Right, because you're looking at rate of change longitudinally, right?
Yeah.
So you really want to know what's the test retest of your own measure.
Yes.
But to estimate the change is bigger in the patient population, I guess, right?
Yeah.
And what's the variation? What I'm asking is, what's the variation in your measure?
I cannot tell you by heart, let's say test retest for PET, but it's very good. I mean, and it's very stable. And when we look at, let's say, tau PET change, the controls that have very tau to start with, the rate of change is very close to zero, and the measures are very similar, let's say baseline versus year 2, and these tracers have been very well validated. So in terms of being able to calculate this, trusting the measure we can get with rate of change, I think this is okay. And I really think this is, for example, for me, because those people are almost not changing, though I think it's very tricky to include them in analysis, or people are sometimes looking at, tau PET in amyloid negative people. I think there we have to be very careful, because the what we're measuring is not binding, so is not capturing so much these fibrillary tangles. So I think we have to, this is a group that we should be careful about.
Thank you.
Is that, okay. And here I grade them out a little bit, actually. So this group is really not changing a lot, and we cannot capture the tau PET level in there is very low. So here we did not analyze them in this group, in this project, sorry. And then if we look in our amyloid positive controls and MCI, we can see the region showing the greatest change is unsurprisingly the temporal lobe, where tau is located. And especially we can see this accumulation being also more prominent now, let's say precuneus, medial parietal. So this is really the spreading pattern of tau PET that we expect. And same, the AD dementia, even though they have a lot of tau already in their brain, you see they're still accumulating a lot and reaching other lobes. The pattern is way more advanced there. So also nice change that we can measure now even in the frontal lobe. And so the first question was to see which measures are more related to accumulation of tau PET over time. And to do that, we ran different regression models. Yes, another question.
Yeah, hi Alexa. On the last slide, maybe it's because you mentioned you grade them out, so maybe that's what I'm seeing. But it looks to me like in your controls, there is a higher rate of change. It's kind of like this purpley blue than in some brain regions for the A beta positive controls. And I was wondering what you make of that.
To those?
Right, because that's kind of like a purplish blue color, which would be higher.
I think it's because I added, I kind of grade them a little bit, because it looks darker. But
okay, but it's actually at 0. I see.
While we are at it, is it normalized, or one, like that 0.1 means 10% increase?
It's the SUVR per year. So
it's 10% increase per year, the red one.
You can see it this way. Yeah. And honestly, I think it's not, and we were talking about this with, like, let's say test retest or something. This is not changing very fast, right? And the pathology is not changing so fast. So I really think here we have, almost 3 years for some participants. So it's getting quite good, at least for what is also available also just in other data sets. But I would say almost maybe 2 years is the minimum, maybe, to start to look at some accumulation, especially in the MCI stage that you start to have more. So still, I think, this rate of change is really relatively small in general. So I think it's also something to keep in mind. But with 2 to 3 years, here we have the quite good sample size. still last year for that, but something to keep in mind, right? It's small changes that we're looking at. Great. So to see, we're wondering, is it more amyloid? Is it more this p-tau 217 that is more related to greater accumulation of tau PET? And so to look at this, we did 2 regression models, across all the brain, two models to look either at the effect of amyloid alone on the left or p-tau alone on the right. And what you see, the colored regions are basically the brain regions where more amyloid or more p-tau is related to subsequent tau accumulation in the brain. And when we look at them separately, you see there's quite strong association with both markers, although it's clear that especially CSF p-tau seems to better track more tau aggregation. And when we put both in the same model, however, we see there's no more effect of amyloid, and it is really greater levels of tau pretty much across the brain that is related to then more accumulation of tau pathology. And this also remains true if we also adjust for high level of tau PET. So even though they already have a lot of pathology in their brain, but still, if we take this into account, our measure, our soluble measure of p-tau 217 still is able to indicate that there will be greater accumulation of tau aggregates. And we try to formalize in this relation, just looking at a mediating effect of tau and amyloid and then subsequent tau PET rate of change. And here you can see there's very strong mediation, as you could already guess from the results on the brain. And here you have the proportion mediation of across all the brain regions. So very strong mediating effect of p-tau 217. So this amyloid-related levels of p-tau are a key marker to be able to track more pathology that's going to accumulate in the brain. And then,
Alexa, I think we have a question in the room. I think I saw a hand.
So here, how do we know this is the amyloid that is mediated by tau and not just tau causing it?
Yeah, that's a very good point. I think we have, again, for this kind of amyloid to tau pathway, we have quite a lot of good evidence. And I think really the main one is that it's really, you need to have, amyloid needs to be present to see elevations in tau.
So this is where I know it needs to be present, but maybe that's just enough. And it's not like the rate of change in amyloid is not related. Like here, you're looking at the rate of change in amyloid, right?
No, this one is, all those two are baseline.
Okay. Okay.
Yeah.
So then the question becomes, if you look at the changes, do they follow as well? How many data points do you have at this point?
For this, I forgot to mention earlier, sorry. And this, we only focused first on these controls in MCI. So that was 130 people at the time.
And 2 PET measures, 2 data points?
Yes, or a bit
so
more.
You can't do longitudinal. Okay, I understand. Thanks.
Yeah, great. And then we wanted to then have some more individualized way to be able to define ROI to look at PET change for subsequent analysis. And the way we went about this in this project was to use connectivity-based measure to look at regions for this, key regions for future tau PET change. And there's been a lot of literature suggesting links between total or functional connectivity. So how the brain, let's say, is wired, or the architecture in the brain can help for the spread of tau. And such an approach has been proposed by Nico Franzmeier, who worked on this with us on this project. And basically the idea here was for each participant, we identify these tau epicenters that we call. They're just the regions with the highest tau load for each participant. And then we have this template of functional connectivity. This was taken from like the controls in ADNI. And we looked for each of these tau PET epicenters, what are the most strongly connected regions? And then if we correlate this overall, the connectivity to the tau epicenters with tau PET rate of change, this gives just an overall measure. This is the y-axis here, an overall measure of connectivity versus tau PET change. And we saw that this whole brain measure, if you will, was also related to CSF p-tau. So people with greater levels of phospho-tau showed, more relation between connectivity and tau PET change. And this was just to prove a bit the idea that this like connectivity can be important. But for the project here, the important was that from these tau epicenters, we know the regions that are, we identify the tau epicenters for each participants, and then we know which are the regions that are more strongly connected to those epicenters. That would be, let's say, quartile 1 to the one least connected to the epicenters, quartile 4. And what we saw is that across all these quartiles, people that were positive on a CSF p-tau, they had greater tau PET rate of change. But basically, this approach here was just a nice way to be able to have some individualized measure to look at tau PET change instead of looking at the whole brain. So for the rest of the analysis, we used tau PET change in this quartile 1. So basically, really, that means from the functional connectivity, these are the regions more functionally connected to the epicenters that are defined at the individual level. And so we have this, and also there's nice work at the moment in press where we try to capture the greatest tau PET change. If we look at just defined ROIs and we look at the same ROIs across everyone, or if we try to take advantage of more individualized measure, I think we can capture a few more percentage of change when we try to go more at the individual level. So we try to take advantage of this in this project.
Oh,
yes?
Alexa, we have a question in the room.
Hi, me, obviously. I don't really understand what this means.
Yeah.
So is it the relation, what's on the y-axis? Is rate of change? Is it a relationship between p-tau and
Here,
number of nodes, or what is it exactly? Is it more?
Yep.
I don't understand myself. I don't.
Yeah. No, I went through this very fast, and it's not critical to understand the rest of what's happening, but I thought it's a nice maybe addition that some people might be interested in. And so, the y-axis here is tau PET rate of change according to 4 regions, so 4 quartiles. And how those were defined was, so for each participant, we identified their tau epicenter, so regions that have the highest tau at baseline. And then we looked from this using a functional connectivity, what are the regions most functionally connected to those epicenters? And this will vary a bit by individual based on their epicenters. And these regions are split based on we split these quartiles based off how strongly they are functionally connected to the epicenter. So the mostly connected regions to the epicenters, the top 25 are quartile 1, then the subsequent one, quartile 2, 3, and 4. And the idea is because we think this functional connectivity is helping tau spreading in general from animal models, or also lots of in vivo data. That was the idea of to say like, okay, so we know where the high tau is, and we know which are the functionally connected region. And this is in theory where tau is going to go next. So if we measure tau PET change in this one, that gives us a bit more advantage than using, let's say, just the temporal lobe for everyone, because people are having the pattern of tau has spread differently by different participants. Is that a bit clear?
Yeah, thank you.
I think there's another in the chat, Alexa. Or maybe
And Aurélie has a question also, I think.
Oh yeah, go ahead.
I think Gabe was first, but
You're here now. Go for it.
okay. So I was wondering how, like, did you look at the overlap between the quartile 1 across your subjects? Like, do they overlap a lot or not? And do they overlap also with the ROI that you used in the previous study that you showed?
Yeah, no, it's a very good question. it's a bit tricky to look at because then that becomes a lot of regions, a lot of parcels. I did not look so carefully how they overlap, I have to admit. Here, the epicenters, these are the real data epicenters across the whole group. If I take the what are the top epicenters across everyone, and it makes a lot of sense, right? So at least that part is, is very, for most people, it's the temporal lobe. That being said, as you mentioned, how it matches with other, regions of interest. So as you saw from the first talk, it's really the temporal lobe, right, that the most tau is. So 95% of people, when they work with tau PET, they use this temporal meta RY, so basically almost the whole temporal lobe. And you'll see all the results I will show. They are using this Q1, but they also hold if we use this temporal meta RY. So, and honestly, so this work that I mentioned will be in JAMA Neurology soon. I think there's a bit of, more, you can get a bit higher rate of change when using some individualized RIs, a bit like quartile 1, but in this paper, we propose something a bit more simple. So I think especially when you start to have MCI patients or maybe AD dementia, then the pattern can be really, really different. And there, I think using individualized measure can be quite nice. You know, if you look at cognitively unimpaired amyloid positive people, like 90% of the time, it's really the temporal lobe. So I think keeping in mind also which population we look at, it can be important. Sorry, that was a long answer, but I hope it,
We have a question in the chat for you from Gabriel. He's asking, why is it called rate of change? Where is time coming in?
This was when we modeled with just the LME in general. This was a longitudinal measure of tau PET, and also then you could see then this become rate of change per year. So all these would be SUVR change per year.
Thanks.
Great. And so now that we have our key individualized way to look at tau PET change, or as I said, right, using just the temporal lobe, you see all the results are the same. And then the next question was, are those measures of tau, either our soluble one or tau rate of change? I should really have said per year. I'm so sorry about that. Is this related to cognitive decline? And we saw, again, focusing first on these controls in MCI only, that both tau measures were related to greater cognitive decline if they have higher levels. And as we mentioned earlier when we were discussing, with tau PET change, it's maybe not so surprising. We knew already this measure is more related to cognition, but it was quite nice actually to see even this soluble measure is related to cognitive decline. And all results I showed also, if we do it separately, only in controls or only in MCI, all results were also significant. So I think, so more robustness to these analyses. And then, so this was for the more prodromal phase of the disease, but this soluble measure of p-tau 217 tends to plateau in more late phase of the disease, so in AD dementia patients. So we analyzed those separately to see maybe there'll be distinct associations. And when we repeat the same, so first, we did not have amyloid PET, in the dementia patient, so that's why it's just CSF p-tau. But what we see is then this time, when we adjust for a baseline tau PET, and we, as you saw earlier, right, they already have so much pathology, we don't see any additive effect or additional effect of a CSF p-tau. So this marker is no longer a good one to be able to track accumulation of pathology in the late stage of the disease. And when we tried to look at this overall association of like tau PET change and connectivity, is this related to CSF p-tau? This was not the case, so unlike what we saw in the prodromal phase. And then when we looked at cognitive decline, where both measures were tracking cognitive decline in the prodromal phase of the disease. This time, no more association of rate of decline with the p-tau measure, but it's really the accumulation of tau aggregates that seems to be a better tracking future cognitive deterioration in the AD dementia participants. And so overall, we see that we have amyloid-related soluble p-tau levels that are an important driver of subsequent accumulation of tau aggregates. especially in the early stages of the disease, but less so in the dementia phase. And both soluble p-tau and the rate of tau aggregation was related to cognitive decline early on, but it's only tau aggregation that seems to be related to more decline in the dementia phase. And then you might be wondering, why are you telling all this about these biomarkers? But if we put these two things together, it could suggest that different forms of p-tau could be targeted at different disease stages when we think of therapeutic options. So in early AD, if we're able to reduce p-tau early on, this might have greater effect to try to postpone cognitive decline. And for example, there are antisense oligonucleotides, one now in phase 3 trial that is targeting this mechanism. So this can be something more beneficial already early on, whereas we think of the late stage of the disease, trying to stop really the accumulation of tau aggregation might be a key pathway to try to diminish the decline in people that might already have them. So by trying to combine, PET data, these novel fluid biomarkers, especially with trying to use longitudinal data as much as possible, we're getting a bit closer to try to teasing apart some mechanisms that could be important to target if we think of disease prevention or a modification.
We got a question in the chat.
Yep.
So question from Alfie asking, just to check I understand, because could this be interpreted as CSF p-tau 217 saturating faster than tau PET, which has a higher ceiling?
Yeah, so that's a good point. I would say not early on. And this is true that then it plateaus in the dementia, but early on it has quite a good range. And I think there are many p-tau markers out there. p-tau 217 is one definitely of the really nice one, really nice range in general. So I don't think that would be the case here.
Thank you.
And I just wanted to mention briefly, I'm not going to discuss this, so I'm keeping this more imaging, but I mentioned these blood-based biomarkers. And for instance, p-tau 217 is also one marker that we can measure very accurately in the blood, and that is showing very, very promising results, similar to the ones that we see with CSF. So really, I think in the near future, when we think of enrolling participants for clinical trials or having less invasive markers, I think this is definitely something that you will keep hearing about. And then, of course, I think, amyloid and tau are key. In AD, they're also at the moment the best biomarkers we can measure. But of course, there is much more to the disease. And we're also very fortunate. We are starting to have much more biomarkers that we can look at and look at different pathways. For example, synaptic dysfunction is really one of the key elements contributing to the disease. Inflammation, of course, vascular dysregulation. So we're starting to have more assays to look at all these things in blood or CSF. And if we take next step, even in terms of having access to a lot of biomarkers, then we can think of omics data that are becoming more and more available. And so, for example, if we take one tissue of interest here, the brain, of course, and we have a lot of data from genetics and so many GWAS studies that have really identified like key gene and key SNPs to look in AD. With transcriptomics data, we can look at gene expression, but more recently, there's also more and more proteomics data, and we're getting closer to actual biology. We can measure of proteins in living humans, so to try to understand a bit more different disease mechanisms. And I'll focus on some proteomics data that we recently have in Lund and that I start to look at. And so this was again done in the BioFinder study. This time we have 982 participants that have proteomics, and we have, as we saw in the other project, those controls, MCI, dementia, but also we have some other neurodegenerative disease or other dementias that we can look into and start to tease apart different pathways, not just only related to AD. And when it comes to the proteomics, we have 2,943 proteins measured in CSF with the Olink technology. And the first analysis that we did was just to look at some group comparisons. So looking at protein differences, again, looking across these AT continuum that we've discussed before. And so we have our group of A minus T minus that don't have much pathology in the brain. Our group that has only amyloid, so A plus T minus, and then a group with both pathologies. And this group then is mostly, dementia people, AD dementia cases. And we have these, non-AD cohorts that I mentioned that we're also starting to look at, but for the purpose of the talk, I'll just focus on this AT continuum. It's already a lot of data to look at. And so first, let's start with this comparison of, negative versus only amyloid. And what you see here is a volcano plot. And so on the y-axis, you have the log 10 of the p-value. Here I used FDR, corrected p-value. So the higher here, the more significant the proteins are. And the x-axis is just the beta coefficient comparing these two groups. So basically here you have the proteins that are upregulated in the participants that have amyloid in the brain versus the controls, and the ones that are downregulated on the other side. And we have the same thing now if we continue on this continuum. So those that have only amyloid versus those that have both amyloid and tau in the brain. And already we can see some key proteins. So for example, MAPT, which is the tau proteins. So that makes a lot of sense and it's reassuring. And also a few key hits. So for example, neurogranin is a synaptic marker that has been shown to be also quite effective in AD. But it's a bit easier to summarize these comparisons with this Venn diagram. So overall, there is actually 13 proteins that were upregulated in both comparisons. They're listed here. And I've put some of the key ones in bold. Some this IT gamma, ITGB2 has been shown in some other work. And when it comes to the proteins, let's say, more related to amyloid, well, as you saw on the volcano plot, most of them are upregulated except ApoE and another one. And when we start to look at more advanced pathology, it's more mixed. You see some, half upregulated, half downregulated proteins. And then, so this can identify some key hits that we can look further into to try to understand what role they might be playing in AD. And to go about this, we looked at 3 different approaches. So first was more this group comparison, but then we wanted to know, are some of those proteins actually related to the levels of pathology in the brain? So are they related to this amount of amyloid and tau directly? And second, with the type of enrichment analysis, we can interrogate whether some specific biological processes are related to those proteins. And so basically with this, we give this list of proteins as input versus a list of background proteins. In that case, in our case, was all the Olink proteins that we measured. And there's many databases where all genes have been annotated for their biological function, molecular function, and so on. So with different databases, we can see, is there some overrepresentation of certain biological processes in the proteins that we identify that are of interest. So that's another way that we wanted to try to understand a bit better these results. And then the last way was to try to look whether some of those proteins are specifically related to different cell types. So using single cell transcriptomics data available, we can see, are some proteins related to certain cell types of the nervous system. So I'll show you some results for these 3 different ways to characterize these proteins.
Alexa, just a quick question for you before you start. So we have Gabriel in the chat asking, did you do a test of the overlaps, rank, hypergeometric overlap perhaps?
Oh, no. And I'm afraid to say I'm not so sure what you mean, but then I'd be very, do you want to talk or? If not, we can.
Hi, sorry, can you hear me?
Yes.
Sorry, I'm in a loud place. So for when you have gene lists where you're interested in whether or not they may have commonalities or differences, there's some statistical tests available to see whether or not what the effects that you've gotten here with those gene lists are actually statistically significant, considering random samples from the gene list. So the one I've used before in a similar kind of vein is rank hypergeometric overlap for comparing ranked ordered gene lists.
Is this available?
There's an R package.
It's an R package. Yeah. Well, I haven't done this specifically. It's just also there's so many of those packages out there. It's crazy. But yeah, I've just used like, you know, keep the terms that were like significant with FDR. But what you're suggesting sounds like way better because you get so many hits. Would you mind maybe putting it in the chat, the name of the link to the package, and then.
Sure, I'll find it and send it along.
Thanks so much. Yeah, no, that's really key. And yeah, I fully agree. I haven't done this yet. So now it's more, yeah, doing the clustering on the non-redundant, like keeping non-redundant terms that, let's say, surviving FDR. But I think, yeah, the approach you're suggesting is really good. So first, yeah, looking at the association with amyloid and tau, and here to focus again a bit more on the prodromal phase of the disease. This was done on all the controls in the MCI participants. And this is kind of a loose term that I've used to maybe orient ourselves with these proteins. So those a bit more related to amyloid, I call them the early, and you'll see they'll be in red. Those that are common across the comparison, I've put them in black, these core proteins, and then I've split the more late proteins if they're like upregulated or downregulated to just help a bit see what we're looking at. And this is a lot. So here's the color that I was telling you about. And basically what you will see on the heat map is the correlation between each of those proteins and the tau when they're in the same model. So then it's easier to see if some proteins are really more strongly related to amyloid or to tau. And we're also interested, especially to look also at tau PET rate of change. So that will be the last column. And with amyloid, actually, we don't see any association with amyloid rate of change. And so here are the results. Again, this is a lot, but I'll just point you to some key aspects. So I think it's already very striking that there are two proteins that are very related to amyloid, very specifically related to amyloid, so SMOC1, and generally also a lot of these more core proteins are the ones showing some of the strongest associations. As we might expect, there seems to be a really key in the AD pathophysiology. But then, as we said earlier, right, it's those amyloid positive people, especially when it comes to tau, that are accumulating, that are showing more tau pathology in the brain. So it was also very interesting for us to show the same type of data, but this time only with positive controls in MCI. And again, we see still, even though people now are already amyloid positive, but we have the same key proteins related specifically to amyloid processes. And what's also quite striking now is we start to see a lot of association with tau PET rate of change. As we said, right, these are now we're restricting to participants that are really accumulating. So then we can, potentially identify some key proteins related to this accumulation of tau aggregates. Next, we try to see about these more biological processes. So here I've used annotations only from this gene ontology biological processes. And this is when I used all protein as input, and we see many hits as expected, but also some that make a lot of sense with regulation of neuronal death, neuronal projections, development, and so on. So many terms, but I think also if I try to zoom in a little bit more, so if I only use the early proteins as input, I found, for example, one hit with cellular detoxification, and these were some of the proteins specifically related to these processes. And we're also trying to look more into protein-protein interaction. And for example, all these proteins are really part of some hub. So trying to use data from different sources, we can find some common pathways or specific mechanisms related to certain key proteins. And lastly, we try to see those proteins, are they related to certain cell types? So to be able to do this, we use the data from the Allen Brain Atlas that released the transcriptomics data from the middle temporal gyrus. And what you will see, the size of the dots then are the proportion of each of those cell types. And you have all the R proteins of interest on the x-axis. Again, it's a lot of dots, but I'll just show you some key aspects. So we were a bit surprised actually to not have more of hits related to microglia, but we have these ITGAM and ITGB2 that were really two of these very strong core proteins. So those are very specific to microglia. SMOC1, that was very related to amyloid, is more expressed in the oligodendrocyte precursor cells. But I think what's very striking also is that the vast majority of these proteins are actually more neuronal, so almost equally represented with either excitatory or inhibitory. So again, trying to can get different information using different types of information to understand better some key hits. And I think I put these little puzzle pieces. It's a lot of data, and then we're still making sense of it and try to identify what are there some pathways that are really key to focus on more in the future and so on. But I think for now, there seems to be a dozen proteins that are very central to the AD pathophysiology. And these 13 core proteins also, if I do the comparison between non-AD degenerative diseases, those proteins are also the same ones are overexpressed in the AD patients. So it's not just neurodegeneration in general, there seems to be quite AD specific. And then some proteins seems to be more related to amyloid, for example, SMOC1, ITGAM, others more related to tau, potentially some also key with tau PET rate of change. And can also identify certain proteins related to either common biological processes and different cell types. But this is a lot of data that we have access to, so there's a lot more we can do. No, I'm exchanges, sorry. For example, looking at the impact on cognitive decline, or there are some really key proteins that together help us identify who's going to progress faster maybe, or trying to find subtypes of participants that have like maybe certain signatures, maybe signatures of resilience, as we were just talking about last week, for example. So we can really start to, look at different biological aspects of the disease and maybe get a better individualized measure of the risk. And some of the key hits we found, for example, SMOC1 also makes sense with the literature. For example, you have one paper here that used both mice data, AD models, or also CSF from MCI. And SMOC1 was one of the key hits of the protein being upregulated, or in CSF, where lots of clustering had been done. For example, SMOC1 was always one of the hub and related to the matrisome extracellular matrix. So also by pooling data from other proteomics data set out there, I think we can identify some key hits that are interesting to continue to look into. And so with that, I'll give you this summary of the talk for now. So the presence of amyloid and tau in the brain, already in cognitively unimpaired individual, was very strongly related to clinical progression over a few years. Along with PET, we also have a lot of new biomarkers to look more into disease mechanisms. And one of the promising one is p-tau217. And we saw that high level of p-tau217 were related to accumulation of tau pathology in the brain, especially in the prodromal phase of the disease. And so this is potentially some pathway we could try to stop if we want to halt cognitive decline. And then looking at the CSF proteome, it reveals many new proteins that are potentially important in pathophysiology that can help us, give us more insight, or maybe new drug targets that we could look into. And with that, thank you for your attention. I look forward to continue the discussion. And it's a big team in Lund, but you have the key people that I've been working with the past year. So thanks a lot.
Thanks, Alexa. As always, like a fascinating talk. And I mean, I haven't seen a lot of your newer research since your postdoc, so it was really exciting to be able to see that.